Hi, and welcome to today's Oxford Centre for Personalised Medicine Flash interview series, where we talk to individuals involved in personalised medicine in one way or another. Today, I'm honoured to be speaking to Dame Kay Davies, Emeritus Professor of Anatomy at the University of Oxford. Hi, Kay. So it's an absolute honour to be talking to you today. Could you just kick off with a brief biography? So journey from undergrad at the university to your current positions and everything in between.
Okay. Thank you, Katherine. So my science career really started with chemistry at Somerville College, which is a 4-year course, and chemistry was fine, but it wasn't particularly inspiring, I'm sorry to say. But fortunately, the 4th year was research year, so I could do the final year in biochemistry. And that's when I really switched towards more biological research. So I did my 4th year in the biochemistry department and then went on to do a PhD. From there, my husband had already gone to France to do his chemistry. We met when we were chemists, and I got a Royal Society postdoctoral fellowship to work just outside Paris. And I chose a lab which is very good at purifying proteins, because I really knew nothing about protein purifications, or really didn't understand proteins because I was a chemist. And it was very fortunately the time when genetic engineering, it seems a long time ago now, was just starting. And so we had to purify our own enzymes, ligase, and so on. You get kits for this now. And I therefore went into the Pasteur to learn some of that technology, because I was working myself outside in Gif-sur-Yvette, about 20 miles south of Paris. By going to the Pasteur, there was a professor there called Bob Williamson, who was based at St. Mary's, who was doing a year sabbatical. And he told me that genetics was the next revolution. And he was right. And would I like to go back to London rather than anywhere else in the UK when we came back, and join his group to set up cloning of disease genes where we didn't know the genetic defect. And so my husband got a job in Oxford, and I indeed went back out to London. I commuted from Oxford to London, actually, for 3 years. And during that time, I was actually funded on a cystic fibrosis research fellowship, but cystic fibrosis was too difficult because we didn't know what chromosome the gene was on. So we decided to start with Duchenne muscular dystrophy as a model disease, because we knew it was X-linked. And we knew roughly where on the X chromosome it was, to develop DNA markers for the first time, to do prenatal diagnosis, first of all, and then that was a way of locating the gene and cloning it. We weren't the first to clone it. Lou Kunkel cloned it first. He was in Boston, still is in Boston. But nevertheless, that really opened up the field. So at that stage, we worked out that all the mutations were heterogeneous, that 66% of patients have big deletions or small deletions. A few of them have point mutations. And so at that point, I was then interviewed for a senior research fellowship by the MRC, and Sidney Brenner, the Nobel Prize winner, didn't have a Nobel Prize then, interviewed me and said, well, you need to be in Oxford, not London, because you waste all this time commuting. So he arranged for me to go and visit David Weatherall in the Institute of Molecular Medicine. And the rest is history, because I moved back to Oxford to be at the WIM. Then for the women scientists bit, I complained to the MRC when my fellowship was coming up for renewal that I was the only person in the WIM, which is an MRC funded unit. They didn't have tenure as a group leader. And so Sir Di Rees at that stage said, right, well, there's an institute in London you can go and direct. It'll give you lots of opportunity. Well, it did give me lots of opportunity, and that was at the Hammersmith. So I then commuted up and down the M40 for a couple of years, but then the government decided to start closing certain London hospitals, and they couldn't make up their minds whether to close the Hammersmith or Charing Cross. And that resulted in planning blight. So I, spending all my time in politics, decided I didn't want to die as a scientist, and resigned and came back to Oxford. And I was very lucky that David Weatherall kept my space. And then when the Professor of Genetics position came up, I applied for it, and I was very lucky to get it. And then I set up my unit, an MRC functional genomics unit, and the Department of Genetics then merged with physiology in the department. So we became the Department of Physiology, Anatomy and Genetics, DPAG now known. And I've worked on Duchenne muscular dystrophy ever since. So I've been working on Duchenne for more than 25 years, trying to find effective treatment for this relatively rare disease.
Brilliant. Thank you so much. So if I say personalised medicine, what comes to mind to you?
The treatment of patients, not necessarily just one patient, but a specific treatment which is tailored to a particular mutation or disease. So in the case of Duchenne muscular dystrophy, it's personalised. If you happen to have a point mutation, it can be treated through codon read-through so that you can get the full length protein. If you've got a deletion that's small enough, we can encourage the machinery to skip those exons that are deleted, and you get a slightly truncated protein, but you get a protein back in frame. So that's what I would call personalized medicine, where you're actually addressing a particular set of mutations.
So could you tell us a bit more about your research on muscular dystrophy, and in particular your work developing therapies?
Yes, so really since the gene was cloned, we've been approaching it in two ways. The first is through trying to encourage people to do gene therapy. This is 20 years ago now, where we noticed, along with many others, that patients had hotspots for their deletion. They weren't randomly deleted throughout the gene. And a lot of them had a very small deletion in one particular end, near the distal end of the protein. But one patient that was sent to me by the late Sarah Bundy turned out to have 50% of his gene missing, which is incredible because he was in his 60s and he needed a stick. And by the time he was 70, he needed a wheelchair. He had a distant relative with the same mutation who was a bodybuilder. And so that patient allowed us to have a muscle biopsy. And we proved that his smaller protein is located at the membrane where you would expect the missing protein to be. And then we cloned his gene and generated transgenic mice and proved that his shortened gene could actually cure the MDX mouse. And so we then said, well, this is the basis for gene therapy in Duchenne muscular dystrophy, because this is a shortened gene. It will fit into AAV, whereas the actual gene is 14 kilobases in Duchenne muscular dystrophy. The mutated protein is dystrophin, and dystrophin is missing in all patients. So you could replace it with this micro gene based on this patient's mutation. And so I'm very pleased to say this year, Sarepta, Pfizer, and Solid Biosciences and Genethon have actually got microgenes that they're taking into clinical trials, and hopefully one of those will get approved this year. So that was founded on work that my lab did a long time ago. But we've also currently been approached a different way. Dystrophin is located at the membrane in muscle. That's the protein that's missing. We discovered another protein which is closely related to it, which we called utrophin, which is expressed everywhere. But early in human fetal development, utrophin is expressed at the membrane and dystrophin isn't expressed. It's a bit like fetal and adult hemoglobin. It's switched off gradually and only found at the neuromuscular junction. That's utrophin, whereas dystrophin is gradually localized more specifically at the membrane as you get close to birth. So we hypothesized that if you could find a way of recapitulating the fetal program, which regenerating muscle does, if you damage your muscle, utrophin comes up again, maybe we could think about curing the disease. So we with our chemistry colleagues, we identified a drug, and with a company which we set up, Summit therapeutics, we identified a drug which did indeed increase utrophin. And we could cure the mouse, or at least prevent the muscular dystrophy in the mouse using that strategy. So we took it into man, and this is a long story, but it's got a good ending almost. We took it into man, and it looked really good after 24 weeks. We got a reduction of regenerating fibers, and the imaging of the muscle looked good. But after 52 weeks, we could see nothing at all. And we now understand that that's because the drug was rapidly metabolized, and therefore, after a certain period, it wasn't effective anymore. So we decided to look, and this is MRC funding, thank goodness, this is long-term funding, which helped. We asked the question, well, what is the mechanism of action of this drug? And we've now found that it targets the AHR receptor, which then indirectly interacts with the utrophin promoter. So, and we looked at other drugs which are antagonists of this receptor, and they also increase utrophin. So we've just now another 2 years funding to try and use them. The medicinal chemists are hard at work trying to find a molecule which doesn't have the toxicity or bad pharmacokinetics that our drug that we took into the clinic did, but still upregulates utrophin. So I'm really hopeful that eventually we'll get to the clinic.
So it's a very, very long process then, I guess,
yes.
going through this.
Yes.
So how are these therapies different and potentially, I guess, superior to other drugs that have been approved or in development for DMD?
Well, the codon read-through, it'll be complementary to that. Exon skipping isn't very effective. So this will hopefully be more efficient than the exon skipping. And with gene therapy, well, gene therapy will probably be better because you're replacing a microgene, but the microgene might not be as functional as a full-length protein. So it'll probably, the gene therapy will probably give you 80% of function back. Maybe if you added a little bit more utrophin on top, a combination type therapy, then you might get an even more effective therapy. So I think it's complementary to what's already out there.
Brilliant, thank you. So I also believe that you've got quite an active interest in the ethical implications of the work that you do. So why do you think this is so important, and what are you doing about it?
Well, initially, it was very important. If you've got a good treatment, and this happened in spinal muscular atrophy, very often if you treat the patients early, you get a better effect. And so the way you can really do that efficiently is to do newborn screening. So I got involved with debating with the Human Genetics Commission, actually. What do families think about knowing in advance that their child is going to be affected by muscular dystrophy? And the answer is not overwhelmingly, but nevertheless in favor of not knowing initially, because if there isn't an effective treatment, there's no point in knowing. Now the scales are beginning to tip in favor of, we've got some treatments, we've got codon read-through. If that gene therapy is approved, I think then neonatal screening for DMD will become a reality and people will want it because they've got hope, whereas before they haven't.
Brilliant. So looking forward then, what do you think the future of personalised medicine looks like? So what do you think the biggest hurdles are, and how might we overcome these?
I think perhaps the biggest hurdle is clinical trials in small groups, because by the time you've subdivided your patients into mutational groups, there aren't very many of them. So I think we'll have to work out effective ways of working with the regulatory authorities to try and design clinical trials with smaller numbers of people so they don't go on for too long, and that we can get proper biomarkers that the regulatory authorities will find acceptable to be able to treat these groups of patients. And that's beginning to happen, but it is a significant challenge. I think the second significant challenge is manufacturing. We've got to make these personalized medicines cheaper, because it's okay for the rich people to be able to treat their families, but these sort of treatments need to be universal. So I think particularly if it's gene therapy, we need to find ways of becoming more efficient at generating the vectors so that we get cheaper product and therefore cheaper drug.
Finally, then, I just wanted to say that you are a huge inspiration to me personally as a very successful woman in STEM. So I just wanted to see if there was anything you'd like to say to the next generation of female scientists coming up through the ranks now. Any words of wisdom?
Well, the first thing I have to remember to say, when I do my life story sometimes, people say, well, that's all negative, you struggle to get to the top. It's a much more favourable environment now for women in science. I think it's a very exciting time for science in general. I think you have to be tenacious, and I think you have to follow your passion. Don't do anything you're not really interested in, because science is always hard. It goes up, there are up weeks and down weeks, and you just have to persevere. But this is perhaps one of the most exciting times in science, so I think there's lots of opportunity, and lots of opportunity for women in particular.
Great. Thank you so much for your time today. It was really lovely talking to you.
Well, thank you for giving me the opportunity.